Wonderful, thank you. Great presentations. Now we're going to pass over to Michael to start his presentation.
All right. Hello, everyone. Okay, first of all, thank you for the kind introduction. I highly appreciate your invitation to this webinar and giving me the possibility to present WACKER Biotech to this audience and show our analytical and manufacturing capabilities for mRNA production. However, I have to be honest, so when Blush asked me around 2 months ago if I want to be part of this webinar, I was a bit hesitant of accepting the invite. The problem was that we just recently purchased a PATfix device, and I was just too worried if we would have enough data to show. However, my worries were unfounded because the device is really straightforward. So it comes with well-developed methods and foolproof SOPs that really minimize your developmental activities. So you can basically start right off measuring your samples. All right, let's start into the talk. In the first part of my talk, I would like to present my company. So many of you may not associate WACKER with life science industry and may rather know WACKER as one of Germany's biggest chemical companies. We have over 16,000 employees, and we are large producers of polymers, polysilicon, and silicones. But we also do have a large biosolutions division and produce bioingredients like cyclodextrins or cysteine, for example, used in baby food. Part of this biosolutions division is WACKER Biotech. This is a 20-year-old contract manufacturing organization with own developmental activities and EMA and FDA approved manufacturing sites. The biopharmaceutical products we produce for our customers, they range from live microbial products to classical vaccines to therapeutic proteins and to products used in advanced therapies, meaning plasmid DNA, mRNA, and lipid nanoparticles. WACKER Biotech globally operates 5 GMP facilities and 1 R&D center. 3 are based in Germany, 1 in the Netherlands, and 1 in US. Plasmid DNA is produced in Halle and in San Diego. RNA and lipid nanoparticles are produced in San Diego, Amsterdam. And in our newest site, also placed at Halle, this is our new mRNA Competence Center, and it opened just in June 2024. And we are really proud to announce that we were awarded the contract by the German government to ensure the production of 80 million doses of vaccines, RNA vaccines, in the event of another pandemic in this site. And in Munich, where I am based, is the corporate's R&D center. Here, 7 different research groups are working on the development of novel technologies for the production of plasmid DNA, mRNA, and lipid nanoparticles. My group, for example, works on the development of downstream processes for both plasmid DNA and mRNA. Okay, so WACKER Biotech is a real end-to-end mRNA CDMO expert. That means we start on gene level. So we assist our customers in mRNA design or also already on template level. We produce the pDNA by microbial fermentation, mRNA by in vitro transcription. We formulate it into lipid nanoparticles, and we have our own fill and finish line. At the R&D center in Munich, we have an own department just dedicated for nucleic acids. We are always staying aware of the IP landscape, which is quite complex actually for the mRNA field, but try to provide proprietary solutions for our customers and offer processes that are freedom to operate where possible. We have our own in-house lipid design and a cell culture lab that ensures that the LNPs we produce are in fact functional, and our customers also appreciate our speed. So we are capable of providing R&D grade material within 1 to 9 months and GMP grade material within 12 months. We also have a good GMP track record. So we produced batches with 80 grams of RNA per batch, and we are, as I already said, part of this German pandemic preparedness program. And we have a wide range of scales. So we are capable of producing pDNA in fermenters of up to 1,500 liters, mRNA up to 30 liters, and lipid nanoparticles in up to 3,000 liters. Okay, so WACKER's corporate R&D has a strong network for advanced therapeutics. We have set up many collaborations with universities like the Humboldt University in Berlin, with the Technical University in Munich. We have even set up a new institute for industrial biotechnology, where WACKER is funding a large number of PhD projects. We are part of several research clusters, for example, CNAT-M for nucleic acid therapy research, or the Saxo-Cell cluster very newly. And we have built up early collaborations with biotech companies like SRTD Biotech or Panthera Therapeutics, where I will show a case study in a few slides. And we assist these companies scientifically, for example, in the design of the RNAs or pDNA, and by providing preclinical material, and this encompasses pDNA, mRNA, and lipid nanoparticles. All right, what are the solutions WACKER Biotech has for the manufacturing of plasmid DNA? We have our own proprietary strains and plasmid, which we term PlasmidTech, and use a cutting-edge USP fermentation protocol that allows us to achieve high yields of plasmid within a fast batch process with titers that can keep up with the leading competitor in the market. Our PlasmidTech strain and the adaptation of the USP protocol already allows high purity of supercoiled plasmid DNA during fermentation, and our cutting-edge DSP process is capable to purify supercoiled pDNA to purities of above 95%. We also work on innovative technologies. For example, we recently patented a new approach for faster and higher pDNA production in E. coli. Or, for example, we have worked on a new approach for plasmid DNA linearization using a CRISPR-Cas9 approach. All right, what are the solutions we have for mRNA manufacturing? We are capable to achieve high yields during IVT reaction, usually in a process that involves post-transcriptional capping. This is also what we suggest to our customers. Sorry, somehow the numbers are a bit off here. But you also don't have to take a loss in capping efficiency for these post-transcriptional capping. So we see very similar capping efficiency. And also our RT-PCR-based method of poly-A length determination shows that the RNAs or the poly-A lengths we produce also match with the aimed size. Our DSP process is capable of reducing double-stranded RNA amounts to below 1%. All right. I don't know what happened to this presentation, but somehow the numbers are a bit off here. Okay, so in this slide, I would like to bring up a success story where I want to show how WACKER's R&D Center efficiently produced research-grade material with high purity for a customer. So we have started a collaboration with Panthera Therapeutics. Panthera is developing mRNA therapies that show tissue specificity by using a combination of 5' UTRs and special LNP formulations. for example, against the acute respiratory distress syndrome. The target mRNA expression protein here is a cartilage oligomeric matrix protein called COMP-ANG1. And we assisted Pantherna by the design of the mRNA and by providing R&D grade material with purities of above 90% and 95% capping efficiency within just 3 months. And Pantherna tested this WACKER process of pDNA templated mRNA production and post-transcriptional capping against a co-transcriptional capping process. And we're able to see that the relative COMP-ANG1 expression in vitro was higher using this WACKER process. And Pantherna was so convinced in our work that they in the end decided to go into mRNA production at our new competence center in Halle, which of course makes us really happy. All right, in the second part of the talk, I would like to put the focus more on mRNA analytics because that's what this webinar is about today. First of all, I want to say that we try to keep our analytical methods up to date with the current guidelines. So what I'm aware of currently is the third draft guideline by the US Pharmacopoeia. I think it came out in August this year. And marked in bold are methods where we now use, in addition, a PATfix device to analyze our mRNA. So what is the portfolio that we have? So for mRNA sequence confirmation, we use a next generation sequencing approach. For RNA concentration, we use UV-Vis spectroscopy or fluorescence. For mRNA integrity, we analyze our samples now with an ion pair reverse phase HPLC chromatography using PATfix, besides capillary gel electrophoresis and agarose gel electrophoresis. mRNA purity, we additionally added a mixed mode HPLC method by the PATfix device to this list. 5' capping efficiency is analyzed by an RPLC-MS method, the 3' poly A tail length by RT-PCR-based method in combination with capillary gel electrophoresis, and residual double-stranded RNA is analyzed in an HT-RF assay that is similar to an ELISA assay, but it's based on FRED. And fragmented RNA, I will later show some data where we also now use this ion pair reverse phase chromatography using PATfix. Residual template DNA is analyzed by qPCR and residual protein by BCA and silver staining. Expression of a target protein is usually at the customer site, but we can assist with our cell culture lab in the development of assays if this customer is still in preclinical phase. And for standard methods like endotoxin or appearance, we use standard pharmacopoeia methods. All right, now to some real-world data. So I would like to do a short excursus to plasmid DNA, and more specifically, so plasmid DNA that is to be linearized as a template for mRNA production. So here we have tested a different buffer composition or different buffer compositions for linearizing pDNA with X-bar1 enzyme. And while agarose gel electrophoresis gives you already a good impression of how the linearization status is, a quantification of linear and circular plasmid DNA is quite cumbersome and error prone. And the PATfix device provides a real remedy here. So we use the CMAC pDNA method to analyze these samples. First of all, in the uncut samples, it's nice to be capable to separate the open circular form and also the supercoiled form. But with this chromatography, you're even possible to separate the linear form from the circular form. And with this, you can easily quantify the amount of linearization within the reactions. So here, for example, for buffer A and number 4, only at this point you have 100% of linear plasmid DNA. And this method is also quite fast, so you get your first result after 10 minutes. And this is, of course, perfect for at-line analytics, where you want to say or want to know at what point is your reaction fully linearized, and you can proceed to save time and costs. Okay, let's get to the next slide. We use the CMAC Prima S HPLC method to analyze our IBT reactions. So on the left side, you see an overlay of Prima S chromatograms, where we took every 15 minutes a sample from an IBT reaction. And you can see that the NTPs are being separated on this column, also your linear plasmid DNA and your mRNA. And you can nicely see how the NTPs are decreasing over time as they are being incorporated into mRNA, and how the mRNA absorbance increases. This method is even faster than the CMAC pDNA method, which is really great to react to these IBT reaction, as it was shown in the video before. So on the right side, you see a function of mRNA amount and NTP amount against time for 2 different RNAs, Cas9 and fluc mRNA. And you can nicely see how after 105 minutes, your mRNA production comes to a halt. And the reason for this is that the ATP is being used up here. So this makes this method perfect for fat batch approaches. So if you would spike some ATP in here, as Tina had shown in the video before, you would be capable of increasing your amount of mRNA in this reaction. In comparison, when you look at the fluc mRNA, you have your NTPs available until the end of the reaction. And this leads to basically a constant increase of mRNA. And I would like to make the point here that it's really critical that you understand your IBT reaction. So each individual component in this reaction has an impact on your kinetics of the mRNA. And you really need to optimize your IBT reaction to each construct. And this is why this PATfix method here is so powerful, because you can easily adapt. All right. We also use the CMAC Prima S method to monitor our DSP steps. In this case, we performed an oligo-DT chromatography run to purify our mRNA. You see your IBT load. So this is the flow-through fraction. This is the wash fraction. This is your eluted mRNA and water. And agarose gel electrophorases and capillary gel electrophorases, they give you a good impression of what the integrity and the quality of the RNA looks like. However, you don't get really much information about the purity of the RNA, especially in terms of small components like NTPs. And these you can see in the Prima S chromatogram. So here, for example, for the IBT load, you have your still remaining NTPs and your mRNA from the IBT reaction. And then in the wash fraction, you see how the NTPs are washed away. A little bit of mRNA is lost also, however, not much. And then in your elution fraction, you have your pure mRNA, which is free of any NTP contaminants. We didn't use a capping reagent here, so if you would have added here, you would also see it in the chromatogram. All right. Then last but not least, we also use SDVB chromatography to analyze our mRNAs. SDVB is an ion pair reverse phase chromatography. And first of all, I would like to show that you're capable of separating your RNA by size with this method. So here for the high range RNA ladder, you can nicely see from 200 to 6,000 nucleotides how the RNA is being separated. Also, our 4 mRNAs we tested, EGFP, fluc, mFix, and Cas9, are separated by size. However, you are not capable of doing exact size determination using this method. And that's why I only show data here for capillary gel lactophoresis, where we see quite well matching results between the theoretical size of the RNA, at least for Fluke and EGFP mRNA. For Cas9, we do see some offset, which we often observe for RNAs that are larger. But we used this method for integrity analysis, and there was a nice comparative study by BS Separations last year in Cell and Gene Therapy Insights, where they compared integrity analysis using SDVB versus CGE. And we used their technique of peak integration, and could see that we had very comparable results between CGE and SDVB analytics. Better for Fluke and EGFP mRNA. For Cas9, we had around 8% difference, which is probably still acceptable. I saw that, so I read this paper again, and I found that they also observed up to 8% difference for their RNAs. For them, it was more or less like an outlier. To say that this is these are the first mRNAs we are analyzing on this method, and we just have to generate more data to see if we observe this more often, or if this is just an outlier here. All right, I would like to sum up this talk. I presented you WACKER Biotech, who is a CDMO expert for advanced therapeutics with over 20 years of experience. We use a cutting-edge analytical portfolio to determine the quality of our nucleic acids. And I showed you how we use PATfix to monitor and optimize our enzymatic reactions like pDNA template linearization and mRNA production, and verify the purification status during these P steps, and determine the integrity of our mRNAs. And at the end, I would like to encourage you that if you're interested in a scientific collaboration project, or need preclinical material, or if you're in search of a very talented CDMO, please just write me. We would be really happy in helping you creating tomorrow's solutions. And with this, I'm at the end of my talk. Thank you very much for your attention. I would like to thank Pantherna for giving us the possibility to show their case study, and I'm curious about your questions. Thanks a lot.